As a CRO, you want to be as excited. You want to be part of that journey with them. And I think that's something that I would say you want to get a sense that these people have a commitment. They want to spend time talking about your product and listening about your product. If they're not willing to do that at the start, they definitely aren't going to be willing to do that later on. So get people that are excited and want to hear about your product and willing to listen. Now, in a practical side of things, there's a couple of things. Everybody thinks their product is unique, but there are some common features of products. So you want to know that you'll spend time and effort looking at the unique part of your product, but there's also an ability to actually, a lot of the routine stuff can be done very quickly and easily. We're not reinventing the wheel for every single aspect of the characterization you need to do for your products.
Are you struggling to find the right analytical strategy and partner for your CMC development? You are not alone. Many biotech teams waste months over engineering methods or partnering with the wrong CRO. Today, I'm joined by Daniel Galbraith, who is the Chief Scientific Officer at Solvias, who has guided analytical development for nearly 3 decades. In part 1, we'll uncover his journey, the biggest industry shifts, and critical techniques for safer cell and gene therapy development. I'm David Brühlmann. Welcome to the Smart Biotech Scientists. Let's dive in. Are you juggling the complexities of CMC development while trying to enjoy the beauty of biotech? Have you ever wondered if there's a way to simplify bioprocessing? Welcome to the Smart Biotech Scientists podcast, where we're diving headfirst into the very challenges you face. We're breaking it down, demystifying the jargon, and giving you the keys to unlock your full potential. I'm your host, David Brühlmann, and I get it. With 15 plus years in the biotech industry, I face the same challenges you do. There's a way to simplify and streamline so you can remove complexity, you can skip trials and errors, deliver without delay your groundbreaking therapy to clinics and market, and still enjoy every single step. Do you want to learn how industry experts and I did it? Grab a cup of coffee and your favorite notebook and pen. Now is the time to take your bioprocessing game to the next level. Let's smarten up biotech. Welcome, Daniel, to the Smart Biotech Scientist. It's good to have you on today.
Thanks very much. Lovely to be here. Thanks.
Daniel, share something that you believe about bioprocess development that most people disagree with.
So you're asking me to be controversial. I like it, just as a kickoff. One thing that I think in bioprocessing, if I look at specifically cell and gene therapy at the moment, everybody seems to think that it will follow the same path that monoclonal antibodies took. 30 years ago, monoclonals, very specialized, difficult to manufacture, and over a period of time, we gradually got better and better, and now we have huge scale, and it's relatively cheap to produce, and we have that all over the world, really. And everybody seems to think that cell and gene therapy is going to follow that same path. I'm not sure that's true. I think we've still got a long way to go before we've got these products that are going to be as scalable and manufacturable, if that's a word, as monoclonals. So I'm a little bit nervous that people are very optimistic about it following the same pathway as mAbs. I think we've got a number of innovative processes to develop before we get anywhere near that. So I'm a little less optimistic than a lot of people on that one.
Well, this is a valid point, Daniel, because advanced therapies are much more complex than traditional biologics. So I think there's things that will translate over, but there are unique challenges that we have not seen in the biologic space. So I'm also curious to see how this will play out.
There's a lot of pressure. You can see that in the market, the conferences. Everybody talks about scalability and making this cheaper, cost of goods, all of that. So there is a lot of pressure. Just don't know if we've got the tools quite yet to be able to get there.
Before we dive deeper into the science, let's talk about yourself, Daniel. Draw us into your fascinating journey from starting your career to now becoming the Chief Scientific Officer at Solvias. What initially drew you into biotech, and what were some interesting pit stops along the way?
Yeah. Interesting. So yeah, long career. It's gone so fast in many ways that you think when people ask you that question, you've been in a long time, you think, not really. But then I started actually in 1996, actually in biotech, which is almost 30 years. Next year will be 30 years. What drew me into it was that mid 1990s period. You started to get the approvals of the first real biologic monoclonal drugs. They were starting to kind of come. The regulators were getting interested, starting putting regulations in place. So it really was that there was an inflection point definitely there where there was these new drugs were coming to the fore. Different challenges. As I said earlier, they were difficult to produce in those days. They were expensive to produce. It was small batches, all of that thing. But they presented a lot of challenges in how to characterize them, how to make them safely. What were the risks? The first time we're making products really in cells. What were the virology risks? What were the bacterial contamination risks? How do we characterize them? So that's what kind of started me off, starting to look at these drugs. It was novelty, it was new, and people are attracted to the new. Along the way, there have been other things that have happened. We started off with them being every product was unique. And then all of a sudden in the mid-2000s, 2005, 2008, we started to look at biosimilars. All of a sudden we could make a copy of these drugs. And then the analytical challenge is with this unique product process, how did we make a copy of that with another product process? So then the analytics started to get more complicated again. This time we were using different technologies to look at these drugs. So again, that was really interesting looking at that different aspect of it. And then more recently, we've started seeing cell and gene therapy. Just around pre-COVID, the COVID time, two things happened. We saw what we could do as an industry around COVID, and I thought that's fascinating. We really put our mind to it as an industry. Sometimes I think we've forgotten very quickly what we managed to do during that COVID period. So it keeps you very interested when as an industry we can rise to that challenge. Along with that, we've seen the advent and the approval of so many cell and gene therapy, mostly autologous drugs, but a lot of drugs that can cure diseases that when I started, we thought we would never have a chance of curing. We sickle cell, all of these kind of conditions now that we've got approved drugs for. That keeps you interested. You can't fail to be interested when you're meeting a medical challenge like that. Now, I'm not on the clinical side of things, obviously, I'm more on the manufacturing side. But you're part of that journey. You're an important part of that journey. And to still be able to at least contribute, even to a small extent, I think, in that journey, it keeps you interested. And it's also, what's next? From when I started out in 96 and 2005, you've seen such huge changes now. And you think, well, another 5 years, where are you going to be now? So you're always looking to the future, I feel. Keeps you young, I hope.
It's evident that you're curious and you've been very excited about all these amazing changes over these last 30 years. What was that spark that drew you eventually into a leadership position and now the CSO of quite a big company? Before we continue, Biotech founders, I want to talk directly to you. Do you know that feeling when an investor asks about your manufacturing strategy and you freeze? One wrong CMC decision can cost you $2 million and 18 plus months of delays. Yet many CEOs are flying blind on these critical decisions. That's why I created the 1-week CMC strategy assessment. In 1 week, you get a personalized assessment of your molecule and development stage. We identify critical gaps before they become million-dollar mistakes, plus a strategic roadmap for high-stakes investor meetings. One CEO told me, David took me from feeling powerless about our CMC roadmap to confidently pitching investors. An absolute game changer for time-pressed CEOs. Whether you are preparing for Series A, approaching IND filing, or facing scale-up challenges, this gives you confidence to ace investor and board conversations instead of surviving them. Ready to stop second-guessing your CMC strategy? Check the link in the show notes and secure your spot. Because your breakthrough therapy deserves a solid CMC strategy. Now, let's get back to our episode.
Yeah, I think you have the scientist role, which is that technical role and understanding what's going on. But I think you need a little bit of that entrepreneurial spirit and that interest in kind of sort of going, well, what's next? What can we improve? What can we make better? And I think that's where the CSO role, I don't think of it solely as purely as a technical role. Obviously, you have to have that technical background because that's really where the vast majority of what you do is. But you need to think, how does it work as a business function as well as a technical function? And I think your job is to inspire. And I think that's really what sort of made me want to move into a more leadership role, is that ability to get people excited about the science, get people excited about what we can do. And I think if you can do that, either with internally with your own company or externally, to match the excitement that these innovator companies have with these new drugs, I think that's what makes you ready to be in that leadership role. And inspiring the science, I think, is what I think of with that. And I'm not saying I'm great at that, but I feel like I can help with that.
And with respect to the cell and gene therapies, as you said, we are now able to treat sickle cell disease and a lot of other diseases we previously thought it would never be possible. What are the specific analytical challenges we are now seeing with these new modalities?
That keeps you young as well. We're trying to tackle some of these challenges. So when we think about these products, we've got cells essentially, and cells are a population. They're not all the same. Essentially, what we're trying to do with the analytics is actually come up with some sort of measure on a population. And that's, for number one, that's an analytical challenge. How do you characterize a population? It's different when you're characterizing a batch of proteins or a small molecule drug like aspirin or something, where everything is expected to be the same. With these products, we expect everything to be different. All of the cells will be slightly different in some way. So we need to sort of like make an average or make a summation of like what's going on there. So as a fundamental level, we have to just think of these products differently. We can't just expect them to behave or to be able to be measured in the same way that we would measure a monoclonal antibody even. So we have to bear that in mind. I think also cells, they don't just have one activity. They do lots of different things. And especially when we've manipulated them. So normally we've inserted a gene or we've modified a gene or something has happened. So we've upset them. We've upset their DNA in some way, usually to make them do something different. And cells will respond in different ways to that. And in some cases, in some of the CAR T therapies, we can see some adverse effects once we've manipulated them in certain ways. So we need to be able to measure that, understand that, what the negative things that could have happened. And sometimes the cells will die, sometimes they'll grow in a particular way. So the summation of all of these changing things mean that the analytical challenges, we have to apply techniques that can obviously make an estimation of what's going on. We need to understand that they'll change over time, because if you take a sample on day 1 of some cells and then look at day 2 or day 3 or day 10, you expect change because the cells will grow or divide or do something. So what are we doing? When are we doing it? And what are the parameters we're putting on there? What are the specifications we want to put on there? And understanding what makes a good batch and what makes a not so good batch really is a big challenge, because normally we're dealing with really small patient batches. When I say patient batches, I mean the number of patients we treat. You might only be making 10 batches for 10 patients, so we don't have a big population of batches to be able to make a specification. So we need to understand that as well. So the challenges are multiple on different levels. What we're measuring, how we're measuring it, when we're measuring it, and then once we have measured it, how are we saying what's good and what's bad?
And one major difference I see also between traditional biologics and cell and gene therapy is in certain scenarios, especially on the cell therapy side, you don't have the classical clinical study progression because you have a patient population of 1, obviously. So how does this play out now when it comes to the requirements with respect to analytical methods? Because usually when we talk about biologics, we have a phase-appropriate approach. Do we still see something similar on the cell and gene therapy side, or how is this adapted?
To some extent, we do. I mean, the difference between what I would call autologous therapies compared to other therapies is really quite important, because you're absolutely right. If we're making one product for a particular patient for treatment, obviously we have to consider that differently to what we would do for a product that we can give to multiple people, all of those sort of type things. But just to take the autologous therapies, because those are the most common ones that we have at the moment that are out there and are approved, we have to understand what the mode of action of the drug is intended to do. So you can just use the CAR T therapies to start with. Essentially, they will express something on the cell surface, and that's expressed molecule on the cell surface. Well, they'll go on and make some interactions and then obviously lead to a treatment that will result with an improvement in the patient's condition. So each batch, as you say, is patient specific. So how do we do that? But essentially, we then look at an empirical level. Do the cells express enough of this protein that we think will be clinically able to achieve a change in the patient that will lead to an improvement? And I think that's where you have to look at historical data, things that are published in the literature, and actually say, if in the past we have seen this level of expression of this type of protein, we will see an improvement there. So for early stage clinical trials, really we're looking at historical data, and that's all we can go on. And I think usually the regulators, they would expect you to be able to look at some other clinical trials that have been published that actually show this level of expression of this particular molecule will be able to achieve the treatment that we're looking for. So I think for these autologous therapies, we're really just looking at just measuring the amount there and then looking back at actually saying, is this good enough? As things move forward and they treat more and more patients, obviously you gather enough data that says when we saw this amount of expression, for example, then we saw an improvement in the patients. And they're gradually gathering more and more information that way, but it's really just a step by step, and they can slowly introduce more specification within it. It's not, I wouldn't say it's in the same way that we would see for traditional biologics, where you have a phase 1 kind of validation, characterization of the assays, phase 2, phase 3, and then commercial release. You don't have that. It's more an iterative process that they have with autologous products, where they gradually see what works and what doesn't, and they introduce it that way. And that seems to be the way that they move with those ones.
We know that in the cell and gene therapy, we have one big challenge. It's the cost, and we have amazing therapies, but only few people can access them today. So my question now, Daniel, is what technologies do you see that help us make better therapeutics faster, cheaper, and perhaps even if we talk about allogenic, perhaps even scale to bigger scales? What do you see coming there?
One of the things I mentioned at the start was that, I think we still need some innovation in this space. I think we definitely do, because from what I've seen, it's a tough nut to crack, the making things to the same quality, to that high specification that we have of safety with the patient. We have seen some products coming through when we see the gene therapies, cell therapies that are using virus to be able to manipulate the cells and actually create the product. Those are quite challenging to be able to scale them on an individual basis, really just because of the vectors that we're using. So I think if we are looking much further in the future, we need to be finding methods that can actually manipulate the cells, that use techniques that don't rely on some of these very expensive vectors that actually are used as part of the manufacturing process. I think we need to sort of look at that. Now, there are some people that are using some of the CRISPR technologies that look at modifying the cells that way, but the efficiency of those, and there are some side effects and some issues around using the CRISPR technology as well. So I think we probably need some innovation in that space that actually looks at a way of actually being able to manipulate the cells in a more efficient way before we can probably scale these. So I think there are a number of techniques where we can scale the amount of cells we produce, and that's been around for quite some time. But to me, it's more in the manipulation of the cells that actually create the gene therapy is actually where we need to focus on that. There are some innovative technologies that look at different ways of manipulating the cells to make them more receptive to the DNA either being inserted or modified or whatever. There are some sort of techniques there, very early stage, not really been taken into the clinic yet. But I think in the next few years, if we can get around that issue, the scaling of the cells themselves shouldn't really be a problem for us, because we've got so much expertise within many companies looking at cells and how to grow them, the media that we use to grow the cells, how we harvest them, and how we characterize the cells is all there. But to me, it's the first step we need to get better at.
And which therapeutic modalities are the most popular right now? Which of those you think will gain momentum, and perhaps other ones are maybe overhyped?
Yeah. If you look across all modalities, I mean, everyone's getting very excited about the peptides and what peptides can do. There's obviously a big push in sort of like that. Looking at the biologic space, the antibody-drug conjugates over the past couple of years seem to have really come to the fore. The opportunities that these are presenting, the payloads, the linker technology has got so much better. There's so much choice now. A lot more manufacturing sites will be able to handle these toxic compounds. And the antibodies, a lot of them are being repurposed into looking at using them as these antibody-drug conjugates rather than just antibodies in themselves. And the other interesting thing we see is that ADCs are being used in combination with other therapies as well, traditional chemotherapies and radiotherapy, all of that, along with biologics. So that's really quite exciting, seeing the combinations of therapies from that point of view. Not too sure I wouldn't say they're overhyped. I think they've still got a long way to go in that ADC, antibody-drug conjugate space. I think financially, there's a lot of companies see that these are something that are commercialized and able to be, there's a lot of return on investment seen there with the antibody-drug conjugates. So that's probably the most exciting in the biologic space. I think overhyped. One thing that seems to have sort of like gone off the boil a little bit is some of the mRNA technologies. It was very exciting through COVID, and obviously seeing the opportunity that these mRNA vaccines seem to have. And that was from a position pre-COVID where you had a couple of companies that were interested in looking at this technology, to everybody all of the large pharma seem to have RNA as a part of their portfolio. That does seem to have sort of like gone off the boil a little bit. Maybe we just need to see a bit more investment, a few more successes in that. It might come back, but it's been a little bit disappointing not to see them come through as actually having a couple of blockbusters to really show what these mRNA technologies can do. And everybody seems to still be working on it, but it's probably been a lot slower than we would have expected to see from what we saw with the COVID and the opportunities over the COVID vaccines and all of that. But I guess now, because we're moving away from the pure vaccine use of these technologies into other treatments, it's maybe taken an unexpected amount of time. I think it was probably a little bit overhyped, as you suggest, to see how quickly some of these could have come through. But those are perhaps the ADCs definitely, like, we see a great opportunity there.
On the podcast, we have covered and talked about choosing a CDMO several times already, but we have never had the opportunity to talk about an analytical partner, a CRO, which also can be, or oftentimes is a strategic partnership. So I would like to know a bit more about that, how to make the best decisions, how to choose the right partner. So let's assume I am the CEO of a biotech startup and I need analytical support because we're lacking certain analytical methods. So when selecting an analytical partner, what are the critical questions I should ask to ensure that the collaboration will be successful?
You make a very good point. This product that you're making as a manufacturer is the most precious thing you have, and you're handing it over to someone in a CRO to analyze it, characterize it, and then produce reports that you then take to the regulators to get approval to go on to your next stage of clinical studies, or else get approval from the regulators to make it a commercial product. So this is a really important, it's your baby, really take things. So you're absolutely right. Choosing the right person to partner from that respect is really important. Now, there's a few aspects that as a CEO that you'll be interested in. One is, can you meet the timelines that we have for getting into the clinic and doing all the things we need to do? So can that partner meet those timelines? Does the company have any expertise? Have they worked with these types of products before? Sometimes you do get very unique products, which, with all due respect, nobody's really got that expertise, but do they have the mindset, the inquiring mindset to be able to work on your product and take an interest in it? And I think that's maybe one of the softer things to ask. And it's more a feeling, do they get excited about your product in the same way that you are? When you describe it, I've been in many meetings where, I love being in these meetings actually, where the drug company is telling you about these patients they want to treat, telling you what this new drug will do and how much it will improve these patients' lives. It's lovely to hear that. And as a CRO, you want to be as excited. You want to be part of that journey with them. And I think that's something that I would say you want to get a sense that these people have a commitment. They want to spend time talking about your product and listening about your product. If they're not willing to do that at the start, they definitely aren't going to be willing to do that later on. So get people that are excited and want to hear about your product and willing to listen. Now, in a practical side of things, there's a couple of things. Everybody thinks their product is unique, but there are some common features of products. So you want to know that you'll spend time and effort looking at the unique part of your product, but there's also an ability to actually, a lot of the routine stuff can be done very quickly and easily. We're not reinventing the wheel for every single aspect of the characterization you need to do for your product. So there's some off-the-shelf solutions that will give me quick and easy answers to some questions that I have. Ask those questions. What can you do for me quickly and what's going to take time? And actually get an understanding from that, because not everything is difficult. Not everything is easy, on the other hand. So where can I save time? Where can I save money by using something's expertise that you don't need to go over any new parts of an analytical technique? There should be a lot of standard things the CRO can offer. And then once you understand that aspect, is what are the resources that you need to be able to put to my product to be able to characterize it, that make it unique, and understand what they can do in that aspect from there. So those are probably things you want to think about. There's some basic stuff, the GMP quality, the quality system, the SOPs, all of the procedures. You want to make sure all of those are in place, but those should really be table stakes. You shouldn't really be worrying too much about that. If you're going to a CRO and you intend to go into an IND, all of those things should be already in place, but you want to make sure they are. Just check out.
That wraps up part 1 with Daniel Galbraith. We've covered his incredible journey and evolving landscape of analytical development. In part 2, we'll tackle the practical challenges from common pitfalls that trip up teams to phase appropriate strategies that actually accelerate your CMC timeline. Speaking of streamlining your CMC journey, check out my new Notion CMC dashboard that will help you transform chaos into confidence. This dashboard also includes a proven map roadmap that will give you the exact step-by-step process that consistently hits IND timelines without the guesswork. And hey, if this episode added value, please leave us a review on Apple Podcasts or whatever platform you found us on. Thank you so much for tuning in, and I'll see you next time. All right, smart scientists. That's all for today on the Smart Biotech Scientist podcast. Thank you for tuning in and joining us on your journey to bioprocess mastery. If you enjoyed this episode, please leave a review on Apple Podcasts or your favorite podcast platform. By doing so, we can empower more scientists like you. For additional bioprocessing tips, visit us at smartbiotechscientist.com. Stay tuned for more inspiring biotech insights in our next episode. Until then, let's continue to smarten up biotech.